All right, so the discussions from the past few modules have provided us with a large toolkit that we can now use to look at specific adaptations that affect human variation. So what we're going to look at in this module is adaptation to high altitude. So this brings up the first question: what do we mean by adaptation? So the term is used broadly and has a wide range of definitions. Simply, it is understood as a category that encompasses changes an organism makes to reduce the negative impact of environmental stressors. And this includes genetic, physiological, and behavioral adjustments. All right, so types of adaptation can be thought of as a spectrum of permanence. So the first level is what we call acclimatization. These are reversible physiological changes induced in an organism when undergoing acute stress. So it is a phenotypically plastic response. So an organism can adjust their phenotype in response to an environmental stressor. So an example is hiking at high altitude. So as one climbs in altitude, one's ventilation rate will increase as the oxygen level decreases. This is a change in phenotype from the base ventilation rate at their normal inhabiting altitude. Once they return to the normal altitude, their ventilation rate will reverse and return to normal. Now, a note about acclimation. Acclimation is essentially the same process as acclimatization. However, it does not occur naturally and occurs in a laboratory setting and when the stressor is artificially induced. Now, another level of adaptation is what we refer to as developmental plasticity. So these are non-reversible physiological changes in an organism when undergoing chronic stress during critical development stages. So again, this is another phenotypically plastic response. So one genotype can produce multiple phenotypes when exposed to different environmental stressors. So an example of this is lung capacity. So individuals born at high altitudes tend to develop larger lung capacities than those who grew up at sea level. So if compared to phenotypes of parents who themselves developed at lower altitudes, offspring will have larger lung capacities than would be expected at random. If the individuals with this larger lung capacity move to lower altitudes, they will retain this phenotype. However, their own offspring at the lower altitude will not inherit that larger lung capacity. All right, and the final type of adaptation is genetic adaptation. These are non-reversible genetic changes and resulting phenotypes in a population when undergoing long periods of chronic stress that results in natural selection. And this is what we're going to focus on in this module. So there are a lot of conditions at high altitude that are going to be impacting an organism. It's typically extremely cold, windy, dehydrating, and there is a high level of ultraviolet radiation. So essentially, it's considered an extreme environment. Furthermore, and more importantly, high altitude environments are apoxic. So this means that they have reduced atmospheric oxygen. And this induces physiological stress as oxygen is critical to metabolic activity in vertebrates. So we have some physiological responses to high altitude. One being hyperventilation. So this is an increase in the rate of ventilation associated with a given rate of oxygen. So native highlanders typically have ventilation rates 40% higher than lowlanders. When lowlanders first ascend to high altitude, they experience an acute increase in ventilation. After a few days, lowlanders acclimatize, which further increases their ventilation rates. Some highland populations have blunted ventilation responses, meaning they have marked lower ventilation rates than acclimatized lowlanders. Now, another physiological response is what we call polycythemia, and this is the increase in red blood cells. Initially, it was believed to be an adaptive response to hypoxia, and the increased number of red blood cells equals more oxygen availability. However, it's hypothesized to actually be maladaptive, as increases in red blood cells results in blood becoming more viscous, which increases the demands on the heart and reduces the ability of blood to flow through narrower vessels, which reduces oxygen affinity. So it is currently understood to be neutral in that the Bohr effect, which is a property of blood pH, balances out the effects of polycythemia, and as a result cancels out the increased viscosity of blood. How do cells detect hypoxic environments? So when the circulatory and respiratory systems fail to keep pace with the rate of mitochondrial O2 consumption, it results in cellular hypoxia. And we have what is called the hypoxia-inducible factor, HIF1. This factor regulates the intercellular response to hypoxia. So it's a transcription factor that regulates gene expression, and it promotes the production of enzymes that are more efficient at using oxygen and proteins that facilitate angiogenesis, which is the development of new blood capillaries. Now, there are two component parts to HIF1. There are alpha and beta subunits. Beta subunits are always in circulation. Alpha subunits are constantly being created and then are immediately broken down. So under hypoxic situations, the alpha subunit breakdown is inhibited, which allows the subunits to join, forming a functioning HIF1 complex. HIF1, being a regulator of multiple aspects of the hypoxic response, is an ideal candidate to study the effects of natural selection on populations at high altitude.